It's my pleasure to be here, and I'm going to talk to you today about accelerating the development of self-amplifying RNA LNP vaccines, kind of a how-to guide for people. And I'm presenting actually on behalf of Danaher Thomas, who's our Director of R&D at PNI and the entire PNI team. So we're very excited to be working in this space. mRNA and self-amplifying RNA-based vaccines and therapeutics are at the front of everybody's minds, especially with the recent successes of Moderna and Pfizer with their mRNA LNP vaccines against the COVID-19 virus. So thank you very much. It's my pleasure, and I'm really honored to be here and working in this exciting area. And I've been doing it for over 20 years. So this has been one of the most exciting years that I think I've experienced during my time in the industry. So again, this is more of a talk about how to use basically the toolkit that PNI is developing. So Genomics Medicines, as everybody is hoping, is the future. And we're seeing the beginning of that, a very exciting time in this area. And Genomics Medicines target and treat disease at its fundamental level. So you can silence genes, you can turn them off with short interfering RNA and other non-coding RNAs. You can express genes using messenger RNA, self-amplifying RNA, DNA, and you can also edit genes as well. So these drugs are designed and not discovered. They have been clinically validated, as mentioned. Manufacturing technologies are ready, and there's a lot of opportunities to really improve human well-being people and treat serious diseases that currently have unmet medical needs. So genomic medicine is a nucleic acid, such as a polynucleotide RNA or a DNA, and a delivery system. So these large polynucleotides are unable to get into their tissues and cells on their own. So you need a delivery system. So work has been done dating back probably well over 20 years on developing lipid nanoparticle delivery systems and variants of those that can incorporate also polymers and various synthetic lipids. This technology is amenable to administration using different routes, obviously with the mRNA vaccines that currently being used for COVID-19, they're using an intramuscular route. They can be administered intravenously for therapeutic applications, subcutaneously, and other routes as well, including oral, nasal, and topical. So again, getting back to the toolbox or the toolkit, this is the PNI's or Precision NanoSystems genetic medicine toolkit. We can help and assist our clients with basically manufacturing the payload, the mRNA or the self-amplifying RNA at the client's direction. They pick the disease target, they pick their sequence, and we can do the rest. We can also help with the delivery solution using our Genvoy delivery platform. So those are lipid nanoparticle based. They're derived from a proprietary lipid library and custom developed into formulations for the given indication that you're going after or vaccine application. So this can be done very rapidly. We have a commercial RUO product called Genvoy ILM. So researchers can start right now with their payload of choice and start doing small-scale experiments to look at the potential of their therapeutics and vaccines. And importantly, the cornerstone and how PNI was founded almost 12 years ago now, 11, 12 years ago, is our nanocellular microfluidic-based manufacturing platform. So this platform is a series of instruments and systems that can make very small-scale formulations, less than 1 milliliter, all the way up to tens or hundreds of liters. So very scalable, very consistent results from early screening and discovery studies all the way up to clinical and eventually commercial development. And again, what makes this possible is the team and the expertise that we've assembled here in Vancouver. So I'm currently based in our headquarters in Vancouver, Canada, And we have, I'm very proud to say, we have a great team, a diverse and very experienced team in drug development and applying these genomic medicine applications. Sorry, wrong button. Okay, so as some of you may know, Precision NanoSystems has now joined the Danaher Life Sciences group of companies, and we have sister operating companies, and I've listed a few here. There's many more: Aldevron, Pal, and Cytiva. And working with these companies, we have a combined portfolio of products and expertise that can design customized workflows spanning from mRNA synthesis, if the client requires that, all the way to fill-finish technologies. So one-stop shop, end-to-end manufacturing workflows. So this integration of workflow integration really requires this expert knowledge of lipid nanoparticles and the unit operations, upstream and downstream. And we found that repurposed technologies and processes are not optimal. So, working with us, we can customize or improve the upstream and downstream unit operations through our process development work and our close collaboration and work with Pal and Cytiva and Aldevron. So it's important to know that from day one, when you're working with this technology, you can go from basically lab bench de-risking all the way through and get consistent results of your particles, consistent performance, physicochemical and biological performance. And for the instrument, for the nanoassembler instruments, they have been, hundreds of these, well over 500 of these instruments have been sold around the world in place in academic institutions, government labs, and also pharmaceutical companies as well. So we are enabling the biopharma companies and, importantly, CDMOs, this nanoassembler technology with no access fees or royalties associated with the actual P&I instruments. So I think that's a really important point. It really helps accelerate the development of clients' programs. All right, so Genvoy delivery platform for developing self-amplifying RNA LNP vaccines. We have, I think, very close to 100 or slightly over 100 different novel ionizable lipids in our library. Working with the client, they'll provide us the disease target, how the target product profile, and we'll design a formulation, selecting appropriate subset of these ionizable cationic lipids into various formulations, and then basically do a screening with the payload, with the self-amplifying RNA or the mRNA. We'll do a screen. Sorry, it popped back. We'll do a screening exercise with the surrogate messenger RNA, what have you, just to show that we have active particles. And you can sort of see the complexity here of the lipid nanoparticle. They're typically in the range of maybe 60 to 120 nanometers in diameter. They can be sterile filtered, and they comprise again of the payload and helper lipids and the all important ionizable lipid that actually enables encapsulation to take place through its interaction with it's positively charged at low pH, the ionizable lipid, and it interacts at that point and binds reversibly with the nucleic acid that you're trying to encapsulate. Encapsulation efficiencies are typically greater than 90% of your payload. Your RNA is fully encapsulated in these particles. So we've also, with the Genvoy platform, we've also shown its applicability for vaccines and gene therapy applications and cell therapy as well. So we have that data. If you want to contact me after the call, we can kind of point you to the data that we have on that. Okay, next slide. So again, this is an example of a luminescence assay with particles, lipid nanoparticles encapsulating a luciferase mRNA or self-amplifying RNA using our Genvoy reagent and I think a published formulation containing the ionizable lipid MC3. We see very similar expression, luciferase expression This is a mouse study after intramuscular injection for mRNA and self-amplifying RNA. In this case, we are dosing at 1/5 of the dose with self-amplifying RNA as compared to mRNA, and seeing similar levels of expression. Interestingly, for self-amplifying RNA, the expression profile is somewhat delayed initially, but it seems like the duration of expression is maintained a little bit longer than for the mRNA, the luciferase expressed in the mRNA formulation or by the mRNA formulation. Okay, so self-amplifying messenger vaccines. Why? Why are we developing self-amplifying messenger vaccines? And a lot of people have said, well, there's already messenger LNP, messenger RNA LNP vaccines approved and in use. Excellent work, very impressive. But we're trying to illustrate, the next step, and the next step potentially is that we could have 10 to 100 times more potent vaccines using self-amplifying RNA. The self-amplifying RNA, without getting into too many details, is enzymatically synthesized in vitro from a DNA template, in many ways similar to the mRNA synthesis. The self-amplifying RNA is then delivered in a lipid nanoparticle formulation to the cytoplasm of the host cell. In this case, for a vaccine, it's an intramuscular injection. The lipid nanoparticles containing the payload have to reach the cell, have to be taken up by the cell, and their payload has to be delivered intact into the cytoplasm to have an effect. So the lipid nanoparticle technology achieves all of that. And there's been a number of papers published probably in the last 10 or 15 years dealing with those mechanisms and how LNP are to be designed to do that, to achieve that nucleic acid delivery. And lastly, for the self-amplifying RNA, once it's in the target cell, it's able to make copies of itself in the target cells and express, in this case, a vaccine antigen and have a potent immune response. Again, a lower dose is very preferable, but also a potential for duration of expression is also very, very interesting and is being explored. Importantly, if you reduce the dose by 10 or 20, 10 to 100 times, you're actually reducing your manufacturing burden, so you can make a lot more doses with less materials and presumably in a shorter time. So that is very good. And also for distributed manufacturing, it's easier and you'll need a smaller footprint if you're designing a facility for manufacturing self-amplifying RNA LNP vaccines. Okay, so PNI's COVID-19 sRNA LNP platform. So back in the end of 2020, the Canadian government, through their Strategic Innovation Fund, funded a number of companies developing responses or vaccine candidates for SARS-CoV-2. PNI was very fortunate to receive this funding, and we've used this funding to develop a self-amplifying RNA LNP vaccine against SARS-CoV-2. And we're currently we've completed a lot of that work to the point now where we have a proven capability to manufacture GMP batches of this mRNA LNP vaccine. And we'll be entering the first half of this year. We're planning to manufacture a clinical vaccine batch and enter phase 1/2 clinical studies for a proof of concept of this technology. Very excitingly, we're not moving. We're still in our current facility, if people know where we are, near Vancouver Airport. But we also received funding from the same funding agency to build a genomic medicine biomanufacturing center located in Vancouver. And we'll have on the second floor of this facility approximately a 32,000 square foot biomanufacturing center where we'll have a full QC lab that will have analytical testing, bioanalytical testing, and also microbiology. So we'll be able to do all of our QC testing in-house to support the clinical batches that are being made in the clean room. So we'll have 2 drug product lines that we are designing and building. The building's complete, and we're in the build out of the of the clean room. That's going to be starting very, very soon and coming online in 2023. So it's a very exciting time for us because we can save a ton of time. At the moment, we work with client projects and then have to transfer them out to CDMOs or to other GMP facilities. And that tech transfer process can take a fair amount of time. Once we have this facility, we can get a client faster to phase 1 clinical trials because all this will happen in-house at one location. Later on, we'll need to transfer to CDMOs as the client progresses through clinical trials and approaches commercialization. But this is a great springboard to get clients quickly into the clinic and hopefully get great clinical results and proceed towards commercialization. So primarily we'll be performing phase 1, phase 2 GMP manufacturing of drug product. Also, we'll have a drug substance, a self-amplifying RNA manufacturing line in this new facility. Okay, so in order to develop a genomic medicine, in this case, a self-amplifying LNP vaccine, core expertise is required and capabilities. So I've just shown here some of the core capabilities that P&I can offer. We typically offer some or all of these capabilities to our clients. It's basically how best we can help the client. So we don't say we have to do it all for you. We'll help you whatever way we can, working with our sister operating companies such as PAL and Cytiva and Aldevron. So please keep that in mind for a very flexible model. Here, these are the core capabilities that you will need. One of those obviously is formulation and process development expertise. Analytical, which is critical. If you're going to make something, you got to know what it is, and developing the appropriate analytical methods is a requirement, not a nice to have. You really need good analytical methods. Underpinning everything is the quality systems and quality control of the incoming materials, the in-process samples during manufacturing, and the final product. So we've built out a pretty impressive quality control team in the last 12 months, and also expertise with technology transfer and manufacturing. Also, project management is super important. So if you start with your target product profile, you know what you want to try to make to treat a certain disease, and then tracking through all the way to GMP manufacturing. So whether that is locking down your formulation or tech transferring to a CMO or a CDMO, your drug product manufacturing process and batch record, P&I can help with that as well. We've completed well over 150 projects for clients, or they're in progress right now. So, we have over 40 dedicated full-time employees working on these contract service projects within my biopharma services team. Okay, some data. So again, we're developing a self-amplifying RNA-LNP vaccine against SARS-CoV-2. And we've used, again, our genetic toolbox, our microfluidics technology using the next gen. And this is some initial data generated with a collaborator. And here we're seeing basically using the toolbox and self-amplifying RNA against SARS-CoV-2, effective humoral and T-cell responses in I believe, a mouse model, looking at different candidate formulations. So we selected 2 lipid nanoparticle formulations for further scale-up work based upon in vitro, promising dose-dependent in vitro and in vivo activity. So they seem quite equivalent. Maybe LNP1 is slightly more potent in vitro. In vivo data is, I would say, more relevant and important. In vitro data is useful, but we have sometimes seen a disconnect between in vitro and in vivo results. So we tend to put more weight on the in vivo data. So we scaled up these 2 formulations, and not to give it away, but we saw differences when we did downstream processing with these similar LNP formulations with the same RNA payload. What we see here is our illustration of our instruments, our next-gen microfluidics technology that's used to self-assemble these lipid nanoparticles. And after particle formation, using either the Ignite, the bench scale system on the left, in the middle, the Blaze system for process development and the GMP system shown on the right, we're seeing very similar critical quality attributes, very similar physical chemical properties of this formulation. So what you make on our bench scale instrument is reflected and consistent as you scale up, which is very good, very helpful in predicting the behavior of drugs during manufacturing. So this is after particle formation. So great scalability. But getting back to those two formulations, LMP1 and LMP2, they're both efficiently induced SARS-CoV-2 specific IgG responses in mice. We're also seeing still that the LMP1 is showing slightly higher IgG response as compared to the second LMP formulation. And they both showed great neutralizing antibodies against SARS-CoV-2. However, when we did downstream processing, I don't have a pointer, but on the left graph, you can see instead of having bars all the same height, after the TFF step for the LMP1, we saw a dramatic increase in particle size. So it would appear that this LMP1 is more shear sensitive. We repeated this a number of times. It's a real result. So I guess the message here is that you can develop a formulation, but you also need to evaluate that in a scaled setting, especially if it's a new formulation, you have limited data. So you really need to demonstrate that as you scale the formulation, it's going to stand up to the process, maybe additional shear, and mechanical stress during downstream processing. Because for TFF, LMP have to go through narrow channels, and they are pumped actively in a circulating system. So that's something to keep in mind. So you need to show that you can scale the given formulation. The process is appropriate for LMP2, but would require further work for LMP1. So we went with LMP2 because we had a solution and they provided similar results. Again, mRNA-LNP vaccines are analytically complex drugs. So you need to have the full suite of analytical testing to completely understand what's going on. So the anchor here, as we've seen, is in vitro potency assay to demonstrate the formulation you're developing and scaling is maintaining its in vitro activity, which is predictive of in vivo performance. But also you need the methods to characterize the actual particle, how well the RNA is encapsulated, where the RNA is located within the particle, LNP composition. Are the lipids concentrations that you put into the process the same as what comes out? Is your nucleic acid to lipid ratio or N to P ratio maintained throughout the manufacturing process? Also talking about payload design, what RNA you're putting into your manufacturing process for drug product. Is it the right sequence? Those things need to be confirmed. And most importantly, and this is an area we're spending a lot of time on working with one of our partner companies, sister operating companies, SciEx, is looking at the RNA quality using capillary electrophoresis techniques. The quality of the input material, and we've demonstrated during our manufacturing process, the quality assessed by CE, the quality of the self-amplifying RNA is maintained through the manufacturing process. And we're tracking that on stability for a number of our formulations that have been made to support GLP tox studies. So, and there's other methods as well that we're looking at for looking at RNA quality. So it's important to have all those methods in place. Otherwise, you're kind of flying blind. You can make something, it looks good when you look at the particle size, encapsulation efficiency, but you need the full set of analytical tests. And P&I can help wherever our clients need that help, the extra help. Okay, this is the last data slide. And for this formulation, we're taking it forward toward the clinic. We did a challenge study in hamsters, and we basically provided the hamsters with inoculated with our experimental vaccine, and then challenged them with virus, with SARS-CoV-2. And we're seeing the vaccine candidates protected hamsters from weight loss. The ones it's the black bar, is the control group, saw significant weight loss. We also protected them from increased lung weight which is most likely fluid accumulating in the lung tissue, protected by both. This is the same formulation with two different candidate payloads, just to be clear. And we also saw higher levels of specific IgG antibody and neutralizing antibody titers. And also looked at some of the look at nasal levels of the rinses and nasal levels, nasal wash levels of excuse me, of SARS-CoV-2 virus levels, and saw a reduction as well in the treated or vaccinated hamsters. So we're very excited about this data. It's a very good model to help us move, and we're moving toward the clinic. And I want to say that this technology is applicable not only to vaccines, infectious diseases and vaccines for that, but also for many other therapeutic applications as well. So P&I really excited to be helping our clients and partners to accelerate tomorrow's genomic medicines from our client's idea to an approved medicine. We have very active in the area of genetic vaccines, working with our clients, gene therapy, and also cell therapy. And I think the future is going to be super exciting. I think the advances, the infrastructure that has been built around responding to the recent COVID-19 pandemic has really increased things like supply chain, the amount of people working in the area as well. And we're identifying actually one of the gaps is talent. So we are actively growing our team, probably like many other companies are in this space. But we need to ensure that we are training the next generation of scientists and engineers that are going to be working with these genomic medicines. So P&I has a lot of intern students from local universities. We're very interested in people joining as a postdoc and working with us, because I think we've taken the kind of perspective that we need to train our own talent because it's hard to find. So it's very bright for the younger generation coming through and using these tools and working in this area. So anyway, I'd like to acknowledge again the Government of Canada for funding. Our collaborators, Dr. Robin Shattock at the Imperial College London and Dr. Yvonne Perry, and the P&I team. And I've listed the people who have been directly involved in generating this data, but I should list the entire company because the amount of collaboration we have at P&I and anything we do is very good. And everyone from the person trying to secure the supply chain for these materials all the way to the person shipping out samples to our clients and to clinical sites is very, it's important. We need everybody, and I want to thank the whole team at P&I there. So if there's any time, I can certainly ask, you can certainly ask some questions and I can try to answer them. If they're more of a biological nature, I might defer those to Anita, who is our R&D director. But yeah, please fire away. Thanks very much for listening, and I hope everybody has a good day.